All right. Just kind of relax in my chair. Morning, everybody. So while people kind of roll out of bed, whatever, uh, anybody have any questions about the stuff that we covered last class? In particular, if anyone did that little problem at the end that I gave, that chi-square problem. If you did, that's cool. If you didn't, give it a shot. It's in the chat session that I uploaded to Blackboard. No, make sure I've got this. Yeah, I'm not mute. Ah. I just checked the weather forecast and it looks like I might avoid getting rained on when I ride home today. Which will be a big plus. All right, so if no one's got any questions about any of the probability and statistics type stuff, get rid of this. Then we can move on with mitosis. So we have two topics to finish up before our first exam, which is next Monday, I believe. Let me just double check. I even checked this morning, shows you what a terrible memory I have. Yeah, so our first exam is on Monday the 15th. And so I would have expected you would be busy revising and studying for that exam already. And if you haven't, then you should start like yesterday, right? Because it will be a challenging exam and you don't have, one would not wish to give up any points in that exam any more than necessary, right? Because if you do badly in your first exam, then you're kind of fighting an uphill battle to kind of increase your average before the end of the semester with the subsequent exams, right? So you really want to do your very best every single exam, and you know, none of them are throwaways. All right, so as I said before, you know, this is a funky format, right? So if you have questions in the upcoming week about anything that we've covered with regards to exam one, if we get time on Friday, we'll talk a little bit about it, but we've already talked about the format of the exams a couple of times already, and we kind of don't have a bunch of time left really to finish the material. But if any of you have any questions outside of class, then please do email me. And if you want to talk, like chat, then we can either set up individual or small group kind of meetings to go over any of the material, right? So that just requires you getting in contact with me. I mean, really, there's only Thursday and Friday, ideally. My weekend's going to be fairly busy this weekend, maybe apart from Sunday-ish. But yeah, if you want to take advantage of that, then get in touch quickly and we'll organize it, right? Don't leave it to the last minute. And certainly don't think about starting to study this weekend because it'll be too late. All right, even if you spend the entire weekend kind of cramming stuff into your head, you know, you benefit from kind of repetition, right? You know, going over the same stuff different ways, different times is how you kind of bake stuff into your brain. Okay, so that aside, what we have today is mitosis and the cell cycle, right? So I'm going to share my screen so you can see, well, there's a schedule, pardon me. All right, we're already going to start around here. So the big deal about mitosis, well, mitosis and meiosis are incredibly important for really who you are, right? So mitosis is really how you get to be who you are, right? From a single cell to your massively multicellular state that you are, right? And thousands of cell divisions. It's also why you age, right? One of the reasons why you age, because your cells have a limit to how many times they can divide, and we'll talk about that a bit later on when we talk about DNA replication. It's probably the easiest place to talk about that. But essentially your cells end up senescing, right? So they kind of grow old and tired and they're unable to keep dividing. And so not only is mitosis and the cell cycle important, kind of taking you from a single fertilized zygote in your mother's womb, but also, and taking you to the person you are now, but also it's important in the decline of your fitness with age. It's also something else that can go wrong, right? And so there's a question to the class, what's the biggest and best example of the cell cycle gone wrong, mitosis gone wrong? Can anybody think? Let me get the chat up so I can see any answers. What would happen if, come on. There we go. Down syndrome, that's an interesting, that's more meiosis. We'll get to that when we talk about meiosis. Not so much mutation, although that can result from DNA replication. Yeah, Sophia's got it, cancer, right? So essentially, cancer is uncontrolled cell division. It's basically restarting development of your body, but in a uncontrolled, unspecified way, right? And so the cell cycle is not just important for controlling, how you develop, but also if it goes wrong, then really bad things can happen, right? And because cancer cells are essentially you, it's very, very difficult for your immune response to, recognize them as wrong, even though clearly, those growths are bad for you as an organism in different ways depending on where they are. So that's why I want to start really talking about the cell cycle, right? So when we talk about mitosis, you have to be careful with language, right? So I try and be as precise as I can be when I use the word mitosis, but it has kind of two different meanings. The first is a very specific meaning, right? And so mitosis in its kind of very most narrow definition is the division of DNA during the cell cycle, during the M phase, right? And so that's this tiny little bit here, right? It's really a very small part of the overall cell cycle and cell division, right? Now, in a more kind of informal sense, we use the word mitosis to refer to cell division, which is essentially results in identical daughter cells, right? So differentiating from meiosis, which is involved in the production of gametes, right, sex cells. And that produces 4 non-identical daughters as a result of the whole process, right? So whenever I use the word mitosis, I'll try and be very specific about what I'm referring to, right? So this PowerPoint is called mitosis, right? And so it really is referring to the whole process of cell division that produces identical daughter cells, right? Now, one thing you probably think is like, well, hang on, we're made up of like lots and lots and lots of different types of cells, which are clearly not identical, right? Skin cells are not the same as liver cells, which are not the same as, muscle cells. They're very, very different. And so that's a different kind of part of a cell's life, right? This essentially what we're talking about just here, this is the birth of a cell, right? This is how cells are born, right? Once they're born, they are identical, right? Because they have the same complement of DNA and the same, more or less, bunch of stuff inside the cells. But once they're born, then depending on what context they're in, like where they're at, what's around them, and so on and so forth, they can then split and develop into different cell types, right? So that's a like a subsequent part. Okay, so when we talk about the cell cycle, we're talking really about the whole deal, and this is very much for eukaryotes. Prokaryotes are much simpler in many ways, right? So we're going to talk about the complicated stuff because this is interesting, this is fun, and it's relevant too. And so the cell cycle really refers to the whole process of starting, right, the process of cell division, all the way to production of two daughter cells, right? So the cells that enter the cell cycle, right, they could either be ones which have just been born, right? So if you're proliferating rapidly, if you're growing embryo, let's say, or you're a cell tissue which has been damaged, or type of tissue that's been damaged, something like that. Those cells will just go straight back into cell division, right, and just kind of keep dividing. So you get this expansion of numbers. Or they could be cells which are just like hanging around going, whatever. And they get the signal saying, all right, Sonny Jim, you're the one, right? And so they get told to go to leave this non-dividing phase, which is called a G0 phase, or I don't know, stationary phase in a sense, and re-enter the cell cycle, right? Then we go through a bunch of growth. G actually stands for gap, right? Because it was when early biologists, using rudimentary microscopes, like, oh, there's just nothing happening here, right? There's a gap. There's a lot of stuff happening in G1 phase, right? Then we have the S phase where DNA is replicated, and G2 phase where a bunch of other stuff which is needed for the M phase is made, right? So this whole part here, which is kind of in this kind of pale green color, is called interphase, right? So that's the preparation essentially of cell division. That's really getting everything ready to the point where you can divide the DNA and then divide the cytoplasm. And that is mitosis and cytokinesis. That's M phase, right? And so by and large, it looks like the cell's not really doing anything in this interphase, but it's actually doing a lot of very, very important stuff. And it's also the largest part of the cell cycle, right? So this isn't to scale by time because it varies. And also the M phase is actually quite a lot smaller than it's shown. If it was shown actual size, it'd be really hard to write anything, right, in the space. So this M phase part, mitosis, is actually a really, really tiny part in terms of time, right? So I don't know the exact numbers. It varies for different cell types, how long they take to divide. But I think a human cell can take anywhere between a couple of hours to 20 hours to divide, right? So it takes a lot of time, depending on the cell type. Okay, any questions so far? This is kind of mostly just nomenclature, like names and stuff. All right. So the other thing that I want to point out here is that there are three very, very important checkpoints in the cell cycle. And these checkpoints are also present in meiosis. And in many ways, one of them, the spindle assembly checkpoint, is even more important in meiosis, because who said Down syndrome? That was Amber, right? So essentially, Amber, this spindle assembly checkpoint or M phase checkpoint, this is what fails to work properly and leads to Down syndrome. Right, so it's this one here that is absolutely critical for making sure each daughter cell gets the right number of chromosomes. Right. Anyway, I kind of jumped ahead a little bit, but I thought that was a cool thing to bring back to what you said earlier. And so there are, and we'll come back to this too, as when we actually look at the phases of mitosis, as in actual DNA division, because it's a lot more relevant there and makes more sense. Pardon me. So essentially, the other two are all about, in large part, DNA quality, right? So the G1 to S, so this is essentially the one that controls the, that'll be later this class, hopefully. Should be. I think I'm pretty much good on time. Yeah, but the spindle assembly checkpoint makes most sense when we talk about it in the context of where we at. Here we go. Oh no, wrong one. Crap. Metaphase, right? So we'll go into it in a bit more detail here. But yeah, so this G1 to S is basically what controls whether or not the cell continues into the S phase, right? And so this is the kind of the absolute go, no go checkpoint. And really what the cell's looking for two things here. One is, are there any like major issues with DNA, right? Are there, is there any DNA that is broken? Does it need to be repaired before DNA replication happens? Because obviously you don't want to copy, damaged DNA, because you'll essentially put that damaged DNA into both the daughter cells, right? And you really don't want that to happen. And also, does the cell have enough juice to make it all the way through? All right, does it have enough energy and resources? Because as soon as this cell goes into S phase, it's committed. And if it can't make it all the way through, say there's not enough resources to finish replicating the chromosomes, right? Or we can't make some of the proteins and stuff required for mitosis later on the M phase, then that cell kind of hangs up. And if it stays in that kind of arrested state for long enough, it will be killed, or it will be induced to kill itself by a process called apoptosis, right? So this is really critical. So if the conditions are not right, then the cell will just pause, right? It will not go into the rest of interface. I don't actually know what happens to cells which are paused here, right? That's something that I need to kind of read up on. I know a lot less about that checkpoint than I do the others. Okay, so if the cell, gives itself the okay, right? It has sufficient resources, it has good DNA, then it goes through DNA replication or DNA duplication, whatever you want to call it. So we now have, so essentially S phase equals two sister chromatids, which are exact copies. Crap, I can't see what I'm writing because my chat box is too far. And that's for every homologous chromosome. Right, so important language here. Homologous chromosomes are, because we are diploid, we have two copies of each chromosome. We have 23 chromosomes, and in total, we have 46 because we have two copies of each. These are called homologous because they are pretty much the same, but they're not exactly the same, right? So one homologous chromosome you got from your mother in the oocyte, one homologous chromosome you got from your father in the sperm. And so when those two come together, they're more or less the same, right? But they have some differences based on what, combination of alleles your parents had. Now, when we go through S phase, each one of those gets duplicated, and then you get your your crab sticks. I'm trying to do it with my hands. You get your exact copies, which are kind of stuck together, particularly around the centromere, right, the central region of the chromosome, right? Those are called sister chromatids, right? Those will later get pulled apart in M phase, right? That's really important language because that's kind of how we talk about these different parts, right? We're not doubling the amount of chromosomes in DNA replication. We're simply copying each one so we have two identical copies that are stuck together for everyone, right? So the chromosome count stays the same. The amount of DNA, like physical, if you weigh it, that would double, but the actual amount of information stays the same. Okay, does everybody get that? That's kind of a crap, don't have any water. That's a really important point to make. Okay, good. So, and this is also kind of building on, in many ways, the work that you did in GenBio 1, essentially, right, or earlier on. So once the cell's gone through S phase, every chromosome has been replicated. Cell gets a little bit bigger, kind of makes more stuff, essentially. And we get to the G2 to M checkpoint, right? And this is also a very important, checkpoint, because it checks to make sure that all the DNA has been replicated and there are no major issues kind of with the DNA, right? So it's not broken. We don't have any kind of weird structures kind of going on, right? And if that those conditions are passed, then the cell enters M phase, the DNA gets pulled apart, and then cytokinesis happens and you end up with your two identical daughter cells, right? That's essentially the cell cycle. Right. So DNA quality is a real big thing, right, when it comes to cell division, because if you imagine you're essentially the result of potentially up to 1,000 rounds of cell division, right, to get from a single zygote to the the millions of cells that make you, right? So if we didn't have these checkpoints in place, then every time those cells divide, there's the risk that you'll have like some catastrophic mutation, right? And then you just end up essentially like a giant bubbling ball of cells, right, which are not doing what they should be doing. It would be catastrophe, right? So it's really, really important to kind of make sure that the quality of the DNA is good. Now, has anybody heard of BRCA1 and 2? This may be of more import to the women in the class. I'm just gonna, annoyingly, I didn't get myself any water. Right, so BRCA1 and 2 are proteins that are involved in DNA repair and detecting damaged DNA, right? Yeah, Amber, that's it, exactly. And so if these don't work properly, right, and essentially, particularly if both of them don't work properly, right, these checkpoints don't function. I can't remember exactly which one BRCA1 and 2 are involved in exactly, right? So it's not a certainty, but if you're homozygous recessive for both BRCA1 and 2, I think the probability is about 80% that you'll get breast cancer. right? Not 100%, right? And even if it was, it still wouldn't guarantee that this would happen, but it raises it to a very, very high level. Because essentially what happens is that, any damage that, mammary tissue cells may have accrued for whatever reason, right? DNA replication or some kind of external insult, that damage won't get detected, right? And so those cells will essentially kind of break free of these checkpoints, and damaged cells will go through cell division, right? That damage can result in kind of more uncontrolled cell division, then you end up with breast cancer. And so probably the most famous kind of person that relates to this topic is Angelina Jolie, which probably most people have heard of. Tomb Raider, UN ambassador, that kind of stuff. She also had a familial history of breast cancer, so she chose to have a double mastectomy rather than risk. And she also had mutations in both copies, BRCA1 and 2. So she had a double mastectomy rather than risk the chance that she would get breast cancer. So just, it's kind of crazy to think about that you would make a decision like that serious based on probability, but the probability is very high, right? So essentially, if any of these checkpoints fail to work properly, particularly the G1 to S and the G2 to M, that's one of the three things that can lead to cancer, right? The other two being kind of inappropriate growth signaling, which is tied into development, and the other one being defective cell death pathways, right? So the cells that are dividing uncontrollably aren't recognized as such, and they're not killed, right? So if you have that kind of perfect storm of three things going on, then essentially you end up with potentially down the road cancer. So there's a lot of work that goes into understanding, these different aspects of cell biology, because it has a very, very, very big impact on what happens to us as individuals. Okay, so that's pretty much it for the cell cycle, right? I'm going to do a little bit on, how chromosomes are structured, like what they look like, right? And so essentially we talked already about the fact that we have homologous pairs of chromosomes, and that's kind of nicely shown here. We also, this is called a haplotype over here. It's kind of like a funky picture. But also this cartoon on the right is very much not what your chromosomes look like normally, right? It's very much a cartoon. Normally your chromosomes are like, this long, right? During, stationary phase, like when cells aren't dividing. It's really only when you enter prophase, right, M phase, that your chromosomes shrink down to this kind of like stubby sort of structure. And they form what's called heterochromatin, which we'll also talk about later. Now, this is essentially what, a chromosome would look like. Fun fact, the P stands for petite, which is French for small. So the small arm is called the P arm because it's this petite arm. I don't know what Q stands for. I think that was just because it was next to P. But also we have telomeres, right, which are protective caps. We'll talk about those later on in DNA replication. And in terms of cell division, the centromeres are actually really important, right? So they're not a region of the chromosome that kind of looks a little bit different. It's still just DNA, but it also has doesn't have very many genes in there, right? So it has a particular kind of form, right? And when cells are dividing, that area, right, essentially results in the assembly of a bunch of proteins, right? So that particular part of the chromosome is recognized by proteins, and this is where what's called the kinetochore forms. And really, the kinetochore what they are is like an anchor point, right? Anybody follow the news about the large radio telescope in Arecibo in Puerto Rico? I think it's Puerto Rico. Yeah, Puerto Rico collapsed recently, right? Which is a crying shame. Super cool telescope. But anyway, essentially what happened is a cable pulled out of like the big end stop that held the big thing above the radar dish. And essentially kinetochores are the same, right? They're the end stops, right? Where the spindle fibers or spindle microtubules attach, right? So if you think about spindle fibers or microtubules being like cables, the kinetochore is the anchor point at one end, right? At the chromosome end, where those fibers attach. And also this is where a lot of the crazy stuff, and this is something that I just about understand, right? And not in anywhere near the detail which is understood. This is where all the special jazz that controls the M phase checkpoint occurs, right? This kind of complex structure of proteins around the centromere. Okay, anybody got any questions? Crap sticks. Sorry, I'm having to drink. This is my Camelback that I use for riding to work in. Forgot to get myself some water before the start of class. Okay, so next part is we really want to talk about the phases of M phase, like mitosis, where it pertains to division of DNA, right? And part of the reason is because, you know, they're important. And one of them has the M phase checkpoint, but also because we're going to be looking at this in more depth when it comes to meiosis, right? And meiosis is quite considerably more complicated than mitosis. Pardon me. So it really, really pays to understand mitosis. Pardon me. Coffee didn't sit well. Before starting meiosis. It's very much the same as why we talk about monohybrid crosses before dihybrid crosses, right? Because if you understand monohybrid crosses, all the principles are there, right? It's just that dihybrid crosses are more complicated. And it's very much the same for mitosis and meiosis, right? So it pays to understand this first, and then we'll talk about all the crazy stuff that happens in meiosis. And it'll be a wild ride on Friday. So best as you can, prepare for it, right? So read through the PowerPoint, listen to the podcast, if you want to call it, whatever. Just have a look at that beforehand. Otherwise, it will be like, because we only have 50 minutes to cover it. Okay, so essentially there are a bunch of stages, right, in mitosis, right? So I always remember this as PMAT because my name's Matt, right? Or PPMAT if we want to be particular. And so another way to remember this too, I mean, you could use pea pods make awful tarts. I mean, that's, no one's going to argue with that, right? You can also use like I don't know what one would say, other words to describe these stages, right? So prophase and prometaphase are preparation, right? So you're getting all your ducks in a row, literally, if ducks were chromosomes, which they're not. Anyway, you're getting everything ready for pulling apart the sister chromatids. Right, so P for preparation, maybe. Metaphase is where the chromosomes meet in the middle, so there's some M's there. Anaphase is when they get pulled apart, and telophase is where you end up with twins, right? So you end up with identical daughter cells at the end. It's kind of a little bit of a fudge. That's the best word I could think of, but if anyone has a better one, then you're more than welcome to share. All right, so essentially this, and these, some of these are very distinct, right? So metaphase is very much distinct from anaphase, but prophase and prometaphase are kind of like arbitrary lines in the sand, I guess, right? It's really a continuum from when interphase ends, right? When that G2 to M checkpoint gets cleared, to when, at the end you have cytokinesis. And even cytokinesis actually starts happening during telophase, right? Again, this isn't kind of this thing. We just teach it that way because it makes it a little bit easier to understand, right? So essentially, there's a bunch of stuff that has to happen, right? If we think about it big picture, right? So we've got the whole cell ready. We've uh, made a bunch of proteins, right? Our mitochondria have done their thing and divided like little funky bacteria type things that they are. We've replicated our DNA, so we have enough to divide up, give each daughter cell, their complement of DNA, of chromosomes. We've done everything else, right? So really, we're all primed. And the very last thing that one needs to do, or last things, is divide up the DNA. You get chromosome 1, you get chromosome 1, chromosome 2, and so on. And then just kind of like pinch those cells down the middle, if you're an animal cell, or kind of build a wall between them, and go, okay, you're two distinct cells now, right? And so when it comes to the mitosis part of M phase, cytokinesis is kind of a little more straightforward in some ways. Really, we've got to do a bunch of things, right? So we start with a cell that has an intact nucleus in it. Inside that nucleus are a whole bunch of, it's like a bowl of spaghetti, basically, right? And at the end of telophase, we'll have two cells, which have distinct nuclei and bowls of spaghetti inside them. And so we have to go from one to the other. And in prophase and prometaphase, what we're doing is we're getting rid of the nuclear membrane, right? Because it's in the way, right? We're moving our centrosomes, right? If you remember, I talked about kinetochores being the anchors, right? That's one anchor. The other ones are the centrosomes, right? They also do other things as well, which we'll talk about in a sec. So we need to break down the nuclear membrane. We need to move those centrosomes to opposite sides of the cell, right? So we can pull things apart. And we also need to condense those chromosomes down, right? Because if you can imagine, you have a cell full of like these huge strands, like spaghetti all over the place. Being able to accurately kind of align those to the center of the cell and then put them apart would be very, very difficult, right? So prophase and prometaphase really is about getting the stage set so the actual process of DNA division is really straightforward. And then telophase is very much the reverse of prophase and prometaphase. You need to rebuild the nuclear membrane, the chromosomes decondense back to what they're like normally, and that's pretty much it, right? So prophase and prometaphase and telophase are really bookends, right, on either end of the process, which is really all about splitting the DNA apart. Oh, that's a good point. So Amber, the long strands, essentially, actually, I wish I had a, no, I don't have a piece of rope. Let's see if I've got some string somewhere. Woohoo! Actually, it's amazing the crap that I have in my drawers. It really is. Right, so if we imagine that this is a chromosome, I actually use this for DNA replication once. Essentially what happens, right, if this is our chromosome, like so, I can't actually show you how long it is. What happens is it gets kind of wrapped around itself, right? It gets turned into super condensed, what's called heterochromatin. And so essentially, if I can, I'm going to try. Let's try wrapping it around a pencil instead. Right, so essentially what happens is that this chromosome gets, oops, kind of coiled, kind of bunched together. It's like a hair scrunchie, really, if one thinks about it like that. And nearly done. Now you have exactly the same chromosome, right? So the DNA is the information is the same. Forgive the labels at the end, right? But now it's kind of squished and condensed down into a much smaller, compact form. And that's where you get those classic kind of X-shape chromosomes. Chromatin as a word means protein and DNA, right? And so the proteins are what the DNA is wrapped around. So at most basic form, you probably all remember this, right? You have nucleosomes, right? We have, shoot, forgot the bloody word right now. Histones, there you go. We have histones and we have DNA wrapped around them, right? What happens is that wrapping, that gets wrapped and wrapped and wrapped even more so, right? And so we just kind of condense it down. It's like if you took a hair scrunchie and picked a strand apart and unpicked like the fabric in a hair scrunchie, you'd end up with one long strand of fiber. And that's essentially a very good analogy between the two. Yeah, so it's still chromosome. I mean, technically, it's in prophase and prometaphase is actually a pair of sister chromatids, right? So alongside this one, you'll have another one here, right? Because these are copies of each other. After anaphase, those copies get pulled apart, and now you just refer to them as chromosomes. And then telophase, essentially the reverse happens, right? This kind of just decondenses. Look at that, wasn't that pretty? Back out to, our long disorganized fiber. So really it's a dynamic process, right? It goes from a very long kind of thing and then it kind of squishes back, kind of coils up in the scrunchie. And then in telophase, it then kind of goes back to its original form. Does that help? Cool. Yeah, that's a good question. So here we go. So this is the end of interphase, right? So what you can see here is kind of what I was talking about, right? These, all the chromosomes which are replicated, right? They're in the form of chromatin because it's DNA and protein, which is how your DNA is normally. And it's all packaged within this nuclear envelope or nuclear membrane. Right, just outside of that, now you have two centrosomes. These centrosomes are going to produce the spindle fibers that attach to each chromosome, right? And each chromatid, really, if you think about it, right, you're going to have one on either side. So in there. In prophase, essentially what happens is those centrosomes push themselves apart, right, using these fibers. So it's literally like they're pushing against each other, and the furthest they can push each other is the opposite side of the cell from each other, right? So that's how those centrosomes, those microtubule organizing centers, as they're called, end up on each side of the cell. The chromosomes, right, now you can start seeing them as distinct individual chromosomes, right? That process of condensation has begun. And then when we get into prometaphase, now you see it continues even further, right? So we've gone from this bowl of spaghetti, I'd guess, right, through prophase, where they're definitely getting shorter and thicker, to prometaphase, where they're real short, stubby. Right. This is really kind of like the end point of that condensation. So this is another way of showing essentially the same thing, Amber, that I just talked about. Now you can see that the centrosomes are on opposite sides of the cell. The nuclear membrane also disappears. And this is a really, really, really important part, right? Each chromatid has a kinetochore, right? That assembly of proteins, that anchor. And so now that the centrosomes are in position, they kind of like anchor themselves, right, to the inside of the cell membrane, right? They kind of like hold on. And from the other side, they fire out, essentially it's like harpoons, right? They just kind of fire out randomly, like kind of like with their eyes closed, if you imagine, right? Obviously they don't have eyes. And every so often, one of those ends hits a chromosome, right? Or actually hits a kinetochore of a chromatid. And if it does, it sticks. If it doesn't, it kind of gets reeled back in, and then it fires it out again, right? And this process takes a while, right? Because it's not like, it's not like you're aiming or anything. It's just totally random. And it has to go on until you have a spindle fiber. So you see this one on the right? A spindle fiber is attached to each side of that chromosome, or each chromatid of that chromosome. So this one here is good. Pardon me. These ones over here are not. So let's just think, kind of questions to the class. If anaphase were to proceed right now, right? So anaphase is essentially where these chromatids separate and get pulled into other sides of the cell. If it were to occur right now, what would the result be in terms of the daughter cells? Would they get the exact same complement of chromosomes as each other? No, right? Because they don't have spindle fibers attached to both sides. So if you were to apply a pulling force, right, these 3 chromosomes at the top will get pulled to the top. These 2 at the bottom will get pulled to the bottom. Right, that's exactly right, Amber. So we haven't even got to metaphase yet, right? But that's a really, really important part. You have to have a spindle fiber on both sides, right? And the same number, right? Sometimes you can have 2 on each side, which is really weird, but mostly it's just 1. Because, come on, what happens now is that essentially those centrosomes are constantly trying to reel in these spindle fibers, right? And so now if you have a centrosome here, pair of chromatids here, and a centrosome here, and they're pulling like equally, what you'll end up with is the chromosome in the middle of the cell, right? And that has to happen for every single pair of chromatids. Every single chromosome has to be kind of tugged around basically until it's in the middle of the cell, right? And even though at this point there isn't anything like saying middle of the cell is here, right? That kind of comes later in cytokinesis. The result is that that's where those chromosomes end up, because they're being pulled equally from both sides. And if you pull equally on something from both sides, it kind of ends up in the middle between the 2 things that are pulling on it, right? And so this is metaphase, right? This is where all of those chromosomes line up on what's called the metaphase plate in the middle of the cell, right? And so this is where the M phase checkpoint kicks in. And so what the M phase checkpoint is all about, basically, give you some words. We're running a little out of time, but we should be good. The chromatids, oops, crab sticks. I've got to move this chat. Here we go. right? So all the way up until anaphase cohesion, dang that's cohesin, sorry, holds those sister chromatids together, right? So even though they're being pulled on, they don't come apart, right? Separase wants to eat the cohesin, right? So there's another part which wants to destroy that cohesin so that those come apart, but we don't want that to happen until we're ready, right? And so we have another thing, and this is a cool word. Something called Shugoshin, right? Ah, this is where I don't want to spell properly. And so there's another protein called Shugoshin, which in Japanese means guardian spirit, which I think is kind of cool. And what it does is it protects those proteins holding the chromatids together, right? And essentially the M phase checkpoint is basically checking to see if there's equal tension on every chromosome. Are we getting pulled equally from both sides? Until that point occurs, Shugoshin will stop separase from destroying cohesin. Once that point occurs, we go, okay, we're all in line, we're all there. Shugoshin stops doing its job. It gets deactivated essentially. And now separase comes in and it eats all the proteins like Pac-Man, right, between those two chromatids. And because we have tension, boing, they get pulled apart, just like that, right? There's nothing sticking them together anymore. So the spindle fibers are pulling them and they pull them apart. And that's essentially what fails to happen properly in when you get aneuploidy, when you get things leading to Down syndrome and the like, which we'll talk about a little bit more in meiosis.
I have a question. This is Amber.
Hey, Emma, go for it.
Why are the microtubules between each kinetochore, why are they extending toward the metaphase plate? What's the point of that?
So the ones that attach to the kinetochore itself.
Well, the ones that are attached to the kinetochore, I know that they pull back, but the
Right.
ones in between each attachment that are extending to the metaphase plate, what's the point of those?
Oh, like the ones that aren't attached
Yes,
to
it's
chromosomes?
it's every other microtubule. What's the point of
Right.
those?
So part of that is it actually helps kind of push the centrosomes apart, right? So the centrosomes you can see are anchored to the like kind of like behind themselves, right, to the cell membrane. But also there are other ones which kind of push them apart as well, right? So it's kind of like making a rigid structure, which then serves as a basis for pulling on the ones that are actually attached to the chromosomes. Kind of like bracing itself, essentially. So that's what those are there for. Those kind of non-attached spindle fibers. Now, did that answer your question?
Yes, it's for leverage, I guess.
Yeah, kind of. Essentially, it's making like this rigid structure inside the cell, which allows force to be applied to the actual chromosomes themselves. So we're a little bit out of time, kind of ran a little short. So we'll finish this up the beginning of lecture on Friday. So Friday is going to be pretty wild because we have obviously this to finish up, which shouldn't take very long, a few minutes. But also we have meiosis to go through, which is, yeah, solid. So I'm going to be like disciplined and focused, no tangents. There will be time for questions, but I'm going to be just kind of plowing through this. Okay, so read ahead the PowerPoint 6 on meiosis. It will help. All right. Does anybody have any questions before I go? Or before we go? No. All right, everybody. If I see you in lab, I'll see you in a little bit. If not, I'll see you on Friday. Have a great day.